genome instability. But more so, and why I'm here today, is that we also found that in particular, this DNA replication function also acts in mitochondria. So not only in the nucleus, but also in the mitochondria, where, for example, HDR repair is not required or not active. So we really, well, maybe I'll just go here. So again, Alan has introduced me to JEMP1 with the relationship that JEMP1 directly interacts with Rev7, which is one of the Fanconi anemia susceptibility genes. And as we heard very early on in this meeting, Fanconi anemia is also ameliorated, or symptoms is ameliorated by metformin, which is actually an inhibitor of mitochondrial complex 1. But what I'm here today to talk to you about is actually a little bit of a different story. And I hope you can bear with me towards the end to discuss with you some of the overlaps with some of the ASD functions. And I guess also, especially when thinking of children with ASD, diet is a very immediate thinking, and dietary supplements and vitamins and iron supplements and fish oil supplements and whatnot. But also for the general population, I mean, we know that diet becomes a more and more important stress factor. We certainly know about, high-fat diet and this overcharred proteins, for example, the black char in your steak and alcohol. And we know that any of these dietary stresses can lead to obesity and accelerate aging, cause diabetes and heart attack, and also cancer. And we also know that certain genetics can amplify or accelerate these disease phenotypes. What at least I didn't know about originally was that one of the number one comorbidities between all of these diseases is anemia. But it's not any type of anemia. It's a specific type of anemia that is called anemia of chronic disease or anemia of inflammation. And that is the number one anemia that is also seen in hospitalized patients, for example. So what is this anemia of inflammation? So when I think of anemia, I think of iron deficient anemia. So for example, like through malnutrition, right? Like in the third world, when you don't get enough dietary iron in your food, then you get anemia because you don't have enough iron to make red blood cells, which require iron. But this anemia of inflammation or anemia of chronic disease is different. What happens is that macrophages, which are part of your blood, of the blood cells, of the immune cells, they're supposed to take old red blood cells, take the iron out of them, and give it back into the bone marrow for new red blood cells to develop. But they basically become reprogrammed. They don't know what to do anymore. And instead of giving it back to the bone marrow, what they do is that they take the iron onto themselves. And so when you look at a spleen section, you can see these macrophages, which are now brown because they took up all the iron instead of giving it to the bone marrow. And eventually they take the iron and actually spit it out wherever they are, which is, for example, here in the spleen. So they deposit it into the tissue. So it's not so much an issue that these patients don't have the iron for the red blood cell development, but rather they can't use it because their cells are reprogrammed and in not being able to give it to the right cells. So that means that treatment with iron doesn't really help with this type of anemia. It helps to some degree because if you put more in, you do get some reaction, but at the same time, you also can actually make it worse because you'll also make those deposits worse, those iron deposits. And so there is this also unresolved, again, involvement of inflammation. We know it's caused by inflammation, but we don't know how or what or why. And one other, again, sort of like telltale feature of this type of anemia is that there is a big sex bias. Males are much more prone or susceptible to this type of anemia compared to females. But again, why is not understood. So we tested this type of anemia by having mice and treating them or putting them on high-fat diet and treating them with an agent called DMBA, which is a polycyclic aromatic hydrocarbon. Essentially, it's the black char on your steak. And when we treat mice with this type of diet, they do get anemia. So they do get this decrease in red blood cells. When we take mice that have a hypomorphic mutation in a Fanconi anemia gene, in this case, it's ROD51C, and these mice on their own actually have no phenotypes at all. So it's a very mild mutation that, again, without any stress, there is no phenotype. But when we treat these mice with the same diet, we see even an increased amount of anemia. So really nicely consistent with the idea that certain genetics can just amplify effects. But what we initially didn't anticipate is that females did not show this type of anemia. They were protected. So we tried to understand what's going on molecularly, and maybe here just in the context of stem cell biology, just for the stem cell biologists amongst you, just to mention that we see similar trends also with platelets and white blood cells, which actually points towards a hematopoietic stem cell issue. So we think that this type of inflammation really is important in terms of stem cell differentiation, at least in the blood cells. But again, something to consider also in the context of neurostem cells. So what's going on? So we performed RNA-seq between males and females and just looked unbiased. What's the difference? What pathways are differentially regulated between males and females with those treatments? And the pathways that came up was essentially inflammation and macrophage activation, which were much higher in the males compared to the females. So we see anemia and we see inflammation. So we just asked simply, do we see anemia of inflammation? Is it this particular type of anemia? And looked at it pathologically again by looking at spleen sections and looking for these granular macrophages, these macrophages that are blown up with iron. And when we score them, we can definitely see that the male mutant mouse has the most of these granular macrophages compared to female or wild type. And similarly, this is also reflected when we look at the spleens for iron stains. So this blue colors show iron deposits in the spleen, which is most prominent in the male and in the mutant. So we see inflammation. So I'm a molecular biologist, so my question is what, from a molecular point of view, can drive this inflammation? And we, of course, know that genomic instability and genomic replication instability causes inflammation. So there's many more papers and many more evidence that is true. And so we looked at specifically nuclear replication stability using this assay system where we can actually look at single molecules of DNA replication forks. And this fork protection that I mentioned before can be measured by this decrease in this replication tract length because those nascent DNAs are actually being chopped up. And we can see that in the mutant cells, in this blue and pink, we get this fork protection defect in both male and female compared to the wild type where we don't really get this. But what's actually interesting is that in this case, both in the wild type as well in the mutant, we see actually an increased fork protection defect in the females, not in the males. So this nuclear instability does not track with this type of inflammation. So what else could it be? So I already mentioned at the beginning that the Fanconi genes are also, and those proteins are also active in the mitochondria, which is the energy cell or the energy compartment organelle of the cell. So that's outside of the nucleus. And mitochondria also have their own genome that needs to be replicated. And so this Fanconi proteins also assist the replication of this mitochondrial genome. And similar to the nucleus, so these are Fanconi patient cells, when this mitochondrial replication is stalled by mitochondrial damage or any kind of DNA damage, we get this degradation of mitochondrial nascent DNA. So we had to develop this new assay system, which we call MIRA, that allows us to really track mitochondrial nascent replication. And so again, we see this degradation of this mitochondrial DNA, which we can rescue with a small molecule inhibitor of a nuclease MR11. In contrast to this nuclear genome instability, this mitochondrial DNA-dependent instability causes a different type of inflammation, and that's, I think, really important. It's called the unphosphorylated STAT1 response, which is a subpathway of your type 1 interferon response. So it upregulates a specific 30 genes amongst the 300 type 1 interferon stimulated genes. And it's also for the cancer scientists amongst you, it's the type of inflammation that's actually causing chemoresistance. But it does not induce type 1 interferon alpha or beta, at least initially, nor the NF-kappa B pathway. And one of the signatures of this particular type of inflammation is that the total protein of STAT1 is increased. And instead of the phosphorylation of STAT1, which is your typical type 1 interferon process. And so again, we can reduce this protein levels when we use this small molecule inhibitor of MR11. So could this type of inflammation be important for this anemia? In order to look at that, we looked at the mitochondrial replication instability with this assay system, with this MIRA assay system. And now indeed, we can see in the wild type male cells that they get this degradation of the nascent mitochondrial forks that can be rescued with small molecule inhibition of MR11, but not the females. And that is also more so true even in the mutant RAD51C male, while the female is protected. So this now tracks quite nicely with the phenotype. So we went back specifically to the RNA-seq data to look if this particular gene signature, inflammatory signature, is upregulated. And indeed it is strongly upregulated in the male, both in the mutant as well as the wild type. And I think that that's really important to point out, because if you look at all 300 interferon genes, you won't find any difference between male and female in the wild type situation. But if you look specifically for this gene signature, there is. So again, I'm a molecular biologist, and so the question is, how are males less protected? And that took us quite a long time to understand, but it was reported that estrogen, in contrast to androgen, actually has a role also as an antioxidant. So that's an agent that can help avert reactive oxygen species, which can damage DNA. And indeed, when we measure that directly in mitochondria, where we induce mitochondrial damage, which we can measure with this red stain agent, mitosox, and then in vitro, in a dish, add estrogen to the cells, we can see that this mitochondrial ROS is greatly diminished. So estrogen really acts as a mitochondrial antioxidant and reduces the DNA damage in the mitochondria that way. So because it reduces the mitochondrial DNA damage, it can also rescue the mitochondrial replication fork defect, because now that replication fork no longer stalls. So we can see this mitochondrial fork degradation in the males. So we see this like loss of mitochondrial nascent DNA, but we can rescue it when we add either a mitochondrial antioxidant or estrogen to those cells, really suggesting that this can then inhibit this mitochondrial dependent inflammation that is causing the anemia. So that's all great. A molecular mechanism where I showed you that estrogen reactive mitochondrial replication instability causes this sex bias in the inflammation and likely the anemia of chronic disease. But I'm a molecular biologist because at the end of the day, we want to have the molecular mechanism inform us on how we can treat. And so the question is then, is this really true? Can we use this information also in vivo? So we went back to our mice where in which we induced the anemia, and then just treat these male mice with estrogen, which can again rescue the mitochondrial defect and then the associated inflammation. And indeed, we can rescue the anemia in these mice. Similarly, if we use an anti-inflammatory aspirin, we can also rescue the anemia. If it was really mitochondrial and not nuclear dependent, we should use a mitochondrial specific antioxidant and also rescue the anemia. And indeed, we can also do this. If we treat the mice with a mitochondrial targeted antioxidant, Mitotempo, we can rescue this anemia. Treating those mice with either this Mitotempo or estrogen can also lower STAT1 protein in the spleens and also reduce these iron deposits in the spleens. So to summarize this, again, we have this mitochondrial replication instability that is really causing this inflammation and sex bias, really showing a molecular distinct role of estrogen as a mitochondrial specific antioxidant, and showing different effects of organelle-specific replication instability with new potential treatment angles against at least the anemia of inflammation. But why do I talk about this at a CHAMP1 meeting? Well, because again, I think that the disease is a very good telltale of what's going on. And we believe that CHAMP1 perhaps can also act inside the mitochondria to suppress this particular mitochondrial DNA-dependent inflammation. So why would we think that? Well, as mentioned, CHAMP1 interacts directly with Rev7, with this Fanconi protein, which we know localizes to mitochondria. CHAMP1 actually also has been reported to interact with IMP2L, which is a mitochondrial inner membrane protein. So a direct interaction with a mitochondrial inner membrane protein also puts it right into the mitochondria. General for ASD, we know, of course, that there is this sex bias where males are much more or more frequently affected. ASD is associated or significantly associated with anemia. The STAT1 protein itself, oh,
Q&A? No, she just asked it. Got disconnected. She's still talking. Can we hear her? Can you hear us?
yes, I can hear you.
Okay, cool.
Am I still
Perfect.
sharing?
No, the sharing has been stopped now.
Okay, let
Cool.
me go back.
No Q&A.
Did I just disconnect? I'm not even sure. What was the last thing you said? You were able to see? Hello?
Sorry, come again?
What was the last thing you were able to see?
Oh, Jesus. Scroll down, please.
What, did I talk already about the champ one?
We were seeing this slide.
Okay, perfect. So so the question is just why am I what are the telltale signs? So this slide.
Yes.
So we are, why am I talking about all of this? Is because we believe that CHIMP1, or we hypothesize at least, that CHIMP1 has a mitochondrial function as well in suppressing this inflammation by mitochondrial DNA instability. And so why do we think that? For one thing, because CHIMP1 interacts directly with Rev7, which is known to localize to the mitochondria. And CHIMP1 is also has been reported to interact with this other protein, IMMP2L, which is an inner membrane protein of the mitochondria. So that really places it inside the mitochondria as well, or at least raises the possibility of. So of course, general from ASD, we know that there is a strong sex bias, with the males being more affected or more strongly being affected. ASD is also significantly associated with anemia, and STAT1 protein itself is an ASD-associated protein. And there is a 2 to 5% increase chance of cancer in ASD patients, in general ASD patients. And also the inflammation seen in neurodevelopmental disease, the types of inflammation or the spectrum really overlaps with that of anemia of inflammation. So we have IL-1 beta, IL-6, and MAP kinase dependent inflammations that really drive this type of disease. So that brought us we're really interested in studying CHAMP1 in the mitochondria. And this is very, very preliminary, but encouraging nevertheless. We did obtain some of the patient CHAMP1 fibroblasts and tested them in our mitochondrial instability assay. And indeed, we can, when we induce mitochondrial DNA stress with this agent called DDC, we see a significant difference between fibroblasts that are from affected patients compared to those that were not. So we really would love to take this assay system into neurons and neuronal progenitors and understand this mitochondrial dependent inflammation in this context, and its suppression in mice specifically, or the outcomes in terms of the neurodevelopmental phenotypes. So with that, I would like to thank the people who worked on this particular story, which is Sunita Roy and Yue Chen, as well as Karl-Heinz Tomaszewski. as well as all collaborators, and Jeff for inviting me to this meeting.
Thank you very much. I don't know if you can
Thank you.
hear, but the people here are cheering for you. So
Thank you.
questions.
So I can't hear any questions. I can only hear
One
you.
second, I'm moving on
Sure.
around, and I will be there in a second.
Oh, okay. I just have a question about so I know you mentioned CHAMP1. Have you looked into POG-Z as well? I know I just found out I have the POG-Z gene, but I also have the iron deficiency anemia. Have you, I know you said females are less likely or are less than males to.
So
Okay.
what's really, we haven't looked into POC, but I certainly listened when somebody in the audience mentioned that they see all this cytoplasmic or cytoplasmic POC. So no, number one, no. And then the other point was the whether females are more protected. Females are more protected if they have the estrogen. So in Fanconi anemia, for example, we know that the patients for sure have endocrine issues. And so in our full-blown mitochondria Fanconi anemia mouse model that really gets all the phenotypes from the disease, we actually don't see any difference between males and females. And so we don't know whether they have, or we actually think that they don't have all the estrogen and all the right hormone balances as a quote unquote wild type mouse, or as a a patient who wouldn't have those mutations. So I think, I mean, clearly females are affected by autism and by neurodegenerative diseases and whatnot. They're less affected, but I think again there's a lot of hormonal questions that also need to be resolved or looked into, especially when you think about sleep issues as well, right? I don't know if that answered your question. Sorry.
I don't actually have a question, just kind of a comment. Dr. Katharina, I was the one that asked about the anxiety earlier, so we got your
Yes.
email. Thank you so much for that. But I just wanted to make just a kind of a thank you to all the doctors and researchers that presented today and those that will present tomorrow. I don't know if I speak for all the parents, but maybe some of them. My high school biology class from 30 years ago didn't really prepare me for what I heard today. But even though most of what you shared was way over my head, it is extremely encouraging, just as a parent, to know that there's brilliant minds at work in this field is very encouraging. And while there might not be, I hate to use the word cure, but there might not be a cure in our children's lifetime, just that you're at work to hopefully improve the quality of life, and certainly for those that would come after. It's very encouraging, and I just would pray that you don't grow weary in your work as maybe weeks and months go by when you don't feel like you have a breakthrough, that that you would stay curious and keep pursuing, and that you would find fulfillment in your work. So we thank you very much.
Thank you. Thank you so much. Again, I'm so sorry that I can't be there, because this is exactly the motivation that that keeps on driving me as a researcher personally, to talk to the patients and make those connections. So thank you.
Hey, have you noticed a difference in age as far as the male to female differences? Does it matter how old they are, or is the difference
It's a
pronounced
very good,
from the beginning?
it's a very good question. So in our mouse model, we induce this anemia with the diet, right? But in generally speaking, this mitochondrial dependent inflammation, so the STAT1 dependent inflammation, is definitely going up with age. And in aged people, this anemia of inflammation is twofold higher in males compared to females. So in aging people, again, that sex bias, like usually when you talk about anemia and open the textbooks, you actually see that females are more prone to anemia because of period and iron loss and whatnot. But after, reproductive years, so in aging, it's the males that have more anemia. And again, it is this type of this anemia of inflammation that is, sorry, that is presenting itself. So there is definitely an age-dependent factor to it as well, which in the first place, you know, Fanconi anemia is a progeroid disease. So it is an accelerated aging disease. So you can also think about it from this angle, that you perhaps just have increased or accelerated aging, which actually also made me think of this hearing frequency of this earlier talk that we heard, that the CHAMP1s are deficient, or was it POXIs? Whether that's the same frequency of hearing loss in aged people. That was one of my questions. It could be an interesting, you know, correlation.
Thank you so much for that. We will finish strong with our final presentation.
Thank you so much.
Thank you very much. Have a nice day.
Bye.